Awesome. All right, guys, we are recording, so I don't mind you, by all means, questions, talk, that's fine. As we discussed before, let's just not mention any identifiable information. Questions for me before we get going too fast here? Okay, I had some questions on this part of the lab, and I know some of you guys asked during class, and I didn't think I had the best answers for you, and that happens sometimes. Tried to make that clear, though. So that's what I would suggest. I'm not going to mark in this book because I don't necessarily own it. I'm borrowing it. But I tried to email a few of you guys back. How many pieces from this cut? If you go look at the animations here, I think they help a little bit. They're a little vague. There are two pieces, right? So what happens is you have this piece and you have this piece. And you have what we call sticky ends. The discrepancy here is referring to, and watch the animation there and see if this helps. There are what they call sticky ends resulting in the cut. And the other questions in through here, I don't really, personally, I just don't care for the way they're worded. I think they're very ambiguous, confusing, and I'm not going to grade them. What I want, but it's not to say completely discount what we're trying to figure out here altogether, okay? The important thing, what we're trying to figure out here is understanding that the enzyme that we're using, this EcoRL that we're using to cut the DNA strands, it's looking for a specific sequence in the DNA, and it's cutting at that location, okay? And then what happens is because of the way it cuts unevenly on both sides of the DNA strand. So you've got overhang on both sides, right? This side you've got a shorter end and a shorter end and a longer end here, and on this side you've got a short end and a long end here. Okay, and that's again technical term. This animation here uses the technical term sticky ends. And that's really what we want you to get out of that, is understand it's not just a clean cut. It cuts one side a little short and the other side a little bit long. And you end up with two DNA strands, right? Two sides on each strand, but two strands of different lengths and different lengths on each side of the strand. Questions there? Does that help a little bit? Okay. All right. And then go ahead and work through the rest of it. Send me your questions as they come up. Ask questions, and we'll go from there. Okay. All right. What else here? Schedule. If we go look at the schedule, right? So there's not just a ton of time here. Today, by the way, is the last day to withdraw. If you did not know that, that's true for any 16-week class you have here at this college. So I'm not telling you, hey, get out of my class. That's not my message here. My message is, hey, if you got that online history class that you forgot about and haven't been doing anything on, today is the day. Go get yourself out of it. Otherwise, it's a little more complicated after today to try to make any decisions, okay? All right, so schedule, where are we at? Today, we'll finish 13, and we'll get a good start on 14, right? On Wednesday, so that'd be the 21st. We should be able to finish 14. I don't think we're gonna get it done today, and that's okay. We should be able to finish 14, and we'll try to hit high points of 15 and 16, all right? We got two more exams. We'll do one exam finals week. I'm not going to do it. There may be some questions on the last exam from previous exams, and they will be the highest of the high points, right? It's not going to be, yes, it'll be slightly comprehensive, but if you really look at everything we're talking about right now, we're talking about all the chemistry stuff previously talked about. We talk about mitosis, meiosis, we're talking about organelles, so it really does all kind of tie back. There's not a whole lot that's compartmentalized and that we don't do much with later. Okay, so again, two exams left. The last exam will have some, it'll be somewhat comprehensive, but it's not going to be real in depth of all of the old stuff. Right, so we'll talk more about the final as we go closer to it, but we do have one more exam between here and there. So not all of 15, chapters 15 and 16 is going to be on the next exam, but there will be some high points. There's some really good stuff in there. We'll try to demystify that on Wednesday, and what we don't get done on Wednesday, we'll hit on Monday. Let's see, today's the 19th, that'll be the 26th, right? So Monday the 26th, we will finish anything on 15 and 16 we didn't get to on Wednesday, and then we will review, right? Now, so that would put us at Wednesday the 28th of test 4, exam 4. I would use those terms interchangeably. That would put that here, right? And we could, in theory, makes me not a lot of difference. We could, if you look at the calendar here, we're talking, this is next week. And then the week after that, the first week of May, that is the last week of classes, right? So that's the 3rd and the 5th, that is the last week of classes. So what I'm thinking is, what I'd like to do is if we proceed with this, that puts us on Monday the 3rd, we'll hit high points, chapters 18 and 19. And those would be the last topics for the last exam, okay? And that would put us at Wednesday, the 5th, would be review test 5. And I've got to go look at the schedule. I think, so the next week then is finals week, right? So the next week after that, this week of May here, this 10th through the 14th, that is finals week. I have to look, I don't remember off the top of my head, but I believe this time frame on Monday, Wednesday, I believe would be scheduled to take their final on May the 12th. So I'm almost certain our final will be either either be May the 10th or May the 12th. Okay. Now, we're not scheduled to meet on May 10th. During finals week, you only meet one time. You meet your scheduled time in your final. We're not scheduled to meet May 10th. I don't mind being available and answering review questions. That's fine with me. It won't be a formal meeting, but I don't mind being here. Okay. And then if that's, again, I'm almost certain. Let's go look. Let's go look. Let's just remove all ambiguity. Rose State College schedule. All right, so that should, even Bing gets that right. Good job, Bing. All right, so spring 21 schedule, it should be a big PDF. We should be able to Control F and find final exam schedules. That's page 65. All right, can I go to 65 and that takes me right to it? Boom, perfect. Okay, these are the 8 weeks. This is the 16-week schedule. We're here. All right, so we meet. So by the way, if you need to know when your finals are, this is how we know. This is how the instructors know. We're supposed to give on during the week of May 10th through May 14th. This is the schedule we're supposed to follow. Okay. Again, we're not scheduled to do anything on May 10th. That's because if you've got a 415 and a 705, you've got 3 classes stacked back to back. That way we don't do anything on the 10th. It doesn't overlap and interfere with you doing stuff on those other classes. Okay. So that's the reason we're supposed to stick to this schedule. So that means there shouldn't be anything interfering with us taking test 5 on May 12th. Again, I'll be available May 10th. If you don't have anything and you want to meet, I'll be here. Okay, just let me know. But that's that's the time we have remaining. Okay. Questions here? Comments, complaints?
So was the last day of class?
It's the 5th.
The 5th is the last day?
Yeah, the 5th is last day of class. Yeah, because then the next Monday is Monday the 10th, and that's when if you got a 415 and or 705, Monday, Wednesday, that's when you'd be taking those finders, in theory. I can't promise your other instructors will stick to that schedule, but I try desperately to. If you're taking classes at another institution and their schedule does not collaborate with ours, let me know. Right? If May the 12th at 5:40 does not work for you, let me know. Let's do that in advance. Let's get that figured out. Okay. Other questions? So like I said, just the high points of 15, 16, and 18, 19, not going to be comprehensive on those, and there's just not going to be a ton of meat here for that 5th exam, so that's the reason we'll do some high points out of the previous exams. All right, so that's what we're looking at. I will start chipping away at getting more and more of those labs graded. I did pretty good the last couple of weeks, and then I've kind of fallen behind, but I will start chipping away at getting those in. I'll get you some homework posted. We've only got, as I said, we've only got about 40 points of homework assignments left. And then just a couple of quizzes. Yes, sir.
One of my lab assignments says missing, but when I click on it, like you can see the submission in there.
Okay. Send me an email and I'll look at it. Could be I just haven't graded it yet.
Okay.
Send me an email and I'll rectify them. Other questions? Cool. Okay.
Nope. Sorry, one more
That's all right. That's all right. What's up?
Will you post any kind of study review for your final study guide?
Ooh. Sure. Yeah, I'll do that. Yeah, because I don't have anything else. It'll be Monday the 10th because I don't have anything else Monday the 10th. But yeah, I should be able to do that.
Thanks, sir.
No problem. Make myself a note. Post review notes for test 3 or test 5. Okay. Yeah. Yeah, I can do something. Will not promise how elaborate, but I should be able to put something like that. All right. Anything else? Okay. There's not a ton to chapter 13. We finished up here last time and said so between the time of, and what we're going to try to focus on today is try to get a handle on what did science do in about the 100 years between when Mendel had conducted his experiments with the peas and had made some really remarkable determinations. We talked about Mendel's laws of inheritance. What happened between then and when we actually began to really understand that genes are coded specifically on DNA, because Mendel did not understand how inheritance was occurring at the microscopic molecular level. He made observations that were very repeatable, heavily supported by the data that he had. And so between that time frame, and again, what Mendel had found went completely against common knowledge in the 1800s about how inheritance occurred. And I've just copy pasted this, by the way. There's a lot of text there. I've just copy pasted this almost entirely from your text. So just kind of copy down the high points here. You don't need to copy every word, but again, trying to really figure out, okay, how are organisms passing on this information? We weren't sure. Well, a theory prevails then based on the work of Mendel by Sutton and Boveri. They proposed a theory early 1900s that chromosomes are in fact what's spreading hereditary information, what's spreading genes. And they've made observations, they've kind of combined observations that have been published about chromosome migration during mitosis and meiosis, how they are getting sorted. They begin to observe crossovers. And the observation is made that even though your gametes, your male and female gametes, are very different, their chromosome, their genetic, their chromosome payload that each one carries is the same, and they're compatible to be combined with another gamete. So you put two haploid gametes together and generate a diploid zygote. So that's, this theory begins to prevail early 1900s. We're still not entirely sure, and this is not widely accepted right up front. This is just published as a theory, and it's kind of the first recorded, where we start putting this together. What Mendel observed is being passed on through chromosomes. Skeptics, of course, would point out, well, there's more genes, there's more traits that we would be interested in than there are chromosomes in an individual, an organism. Right? We only have 23 pairs of chromosomes in humans. There's only 46 chromosomes. There's way more than 46 traits that appear to be inheritable from generation to generation. So how do we figure that out? Well, work begins starting kind of at this point moving forward, looking at individual chromosomes and the idea that each chromosome is likely carrying genes and carrying information for more than just one trait, more than one gene. Okay? And again, one of Mendel's laws is that these alleles get separated independently, right, and the genes are inherited independently. He had studied 7 genes that they were completely independent as to how they were being inherited. Okay? And so what that means is that there's about a 50% chance as to whether a gamete contains, if we're looking at 2 genes, there's about a 50% chance that those 2 genes are inherited from one parent together, or there's also a 50% chance that one gene is inherited from the mother and one gene is inherited from the father. So there's about, if the genes are truly inherited independently of one another, there should be a 50% chance, whether you're getting one from each parent or a 50% chance you're getting both of those traits from the same parent, if they're truly independent. Now, when scientists really began to dig into this and they started looking at more and more traits, and what Mendel had observed, they did start seeing some traits that weren't completely independently sorted. And they were trying to rectify this. We're trying to figure out, why is that the case? Okay, well, we have what we call this recombination frequency of 50% or 0.5. So again, if they're truly independent, it's a 0.5 recombination frequency. And whether they're from 2 separate parents or from the same parent. So, and your text does a really good job kind of outlining the basics here. And there's a link down here. I've copied the link from your text. There's a graduate student working in a lab trying to figure this stuff out. And the story is, the legend goes, is Alfred Sturtevant, looks like, takes home lots and lots of statistics and just sits down. I mean, he doesn't have Microsoft Excel at home. This is 1913. Right? He can't just fire up his computer, plug it all in, and have it graph it for him. He actually takes home this pile of data and tries to work out how the recombination is occurring. And he starts to notice some patterns between some of the different genes. He notices that, and they're looking at a handful of traits that they have been able to trace back to the X chromosome on some flies, specific species of flies, Drosophila. And he's figured out that the recombination rate occurs differently for each trait. Some traits are more likely to recombine, and some traits are more likely to be inherited directly from one parent. And again, hopefully read through the text, I really think this link, I tried to work out, clicked on this link here, we'll see if it'll load. I tried to work out as like, what's a good way to explain this? And I went and looked at this animation like, yes, this is as good or better than I'm going to do. So I'm just going to refer you to this link. It's also in your text. Okay. Nice picture of Mr. Sturtevant here. And it actually has got some nice graphics in here of, so go through and watch this, right? Click through this, read it. Hopefully this kind of helps a little bit. But he's, it's showing you recombination of the genes and talking about how was it that they figured out that females versus males with these different traits, and they were actually able to, and this is the graphic over here. I'm going to close this out. So again, these are the traits that he's looking at. He figures out the recombination rates of these alleles on the X chromosome. And he's able to figure out that that correlates directly with location on the chromosome. Where on the X chromosome is the information for these different traits coded, stored, and that it's inherited differently based on how close or how far apart those particular alleles are on the same chromosome. And so we coined this unit of measure we call the centimorgan, right? The centimorgan basically says that there's a 1% chance of recombination for every centimorgan of distance along the chromosome that those alleles are stored. So what that means is the more distance between the alleles on the same chromosome, the more likely that there's going to be a recombination in between those locations on that allele. Okay. So for example, in this particular species, they're saying, if you look here, the black body, the body color, black versus gray, right? That's going to be inherited along with the eye color more likely than, say, the body color versus the length of this R stem, or this other eye color down here, brown eyes versus red eyes, right? So it's a long distance between the body color and brown versus red eyes, a little bit shorter distance between body color and wing length. So what he's trying to say is it's more likely that the body color and the wing size are going to be inherited together as opposed to body color and eye color. Those are, because that's more than 50 centimorgans between each other. That's a 0.5. That's more than 0.5. They're almost guaranteed to be unlinked. So if it's 0.5 or more, those traits are going to be inherited independently. 50% is recombinant and 50% parental. But if they're closer together, if they're less than 50 centimorgans apart, then there's a possibility that they're actually going to be linked together. They're less likely to be separated or recombined independently of each other. Okay. Now, if they're on different chromosomes, if the traits are on different chromosomes, then they're going to be inherited independently. It's only traits that are on genes that are on the same chromosome this becomes a factor. And again, the reason that they were able to provide definitive proof of this is because they're looking at traits only on the X chromosome. So it only needs, in the males, it only needs one trait to be exhibited. It was a little bit easier for them to track. Okay. All right. Questions about this? I know that's really abstract. I know this one's out there a little bit. Okay. But again, it's just understanding that the closer on a particular chromosome the traits are, the less likely they are, the more likely they're going to be inherited together. So if the mother, if the mother has short wings and this particular variation in their eye coloration, okay, because there's less than 10 centimorgans between them, most likely if the mother has these traits, the the mother's going to pass that along. The maternal gametes are going to, these two traits are going to be tied together. It's unlikely that they're going to swap between the maternal and paternal X chromosomes between that short of a distance, as opposed to this longer distance. Those are going to get swapped in between, almost guaranteed. Okay. All right. Another thing we talk about chapter 13 is we talk a little bit about chromosomal disorders. What happens when this doesn't go quite right? So there are situations where we have what we call a non-disjunction. A non-disjunction is where something gets messed up in the separation of chromosomes during meiosis. And so you have a gamete that has the wrong number of chromosomes, either not enough or too many, right? So in this case, during mitosis, or sorry, meiosis 1, you have one pair of homologous chromosomes that don't separate, right? So now in meiosis 2, you've got this set of gametes is going to have an extra chromosome, and these are going to have one less chromosome because this pair ended up going this way, and the cells that resulted this way didn't have the extra pair from over here. Okay. You can also have situations where you can have, during meiosis 2, you could have the separation does not occur correctly here, where one gamete ended up with an additional chromatid, essentially, but in this other direction, they're just fine. Okay. So again, a couple ways that can happen. What results of that? Well, most of the time, what we find is the larger chromosomes. Okay, so again, there's 23 pairs of chromosomes. There's 22 autosomes and one pair of sex chromosomes in humans. What we normally find is if an individual has the wrong number of chromosomes, right? So you have a gamete that has the wrong number of chromosomes, combines, fertilizes with a gamete that has the right number of chromosomes, the resulting zygote has the wrong number of chromosomes. Maybe they have one too many chromosomes. That's trisomy, where you have 3 of a particular chromosome. It could be more, actually. You could have monosomy, where you have an individual with 1 too few chromosomes. Most chromosomes, this is fatal. Most chromosomes in most organisms, this is fatal. At least in animals, right? Animals, especially in mammals, this is pretty fatal most of the time. There are a few exceptions. But your larger chromosomes, as I was mentioning earlier, we've got 22 chromosomes, 22 autosomes, and 1 pair of sex chromosomes in the human body. Most of the autosomes, especially the larger ones, if you end up with 1 too many or 1 too few of the large ones, because there's so many genes coded on the larger chromosomes, it does not work out well. It just simply doesn't work. Some of the smaller ones we find, though, it's survivable, but most likely the individual is going to have some type of disorder as a functionary. Okay. And how do we know this? How can we prove this? Well, one way we can prove this is through what we call a karyotype. A karyotype is where we take a microscopic image of a cell's chromosomes right at the point of mitosis. Okay? Right at the point of mitosis, right as these things are getting ready to move to the metaphase plate, be separated. That is the one instant in time where you can visibly see under a microscope all the chromosomes and their structures. Again, most of the time, those chromosomes aren't all spooled up and contained neatly packaged. But during mitosis, we can physically see these chromosomes. What we can do is we can take a snapshot. We can take a picture on our microscope. And then we can take that digital image and cut, copy, paste pieces of it. You can crop out a little piece, right? Go into Photoshop or whatever software you're using. And grab this guy, right? And that looks like, which one is that? It looks like this. we've already established I'm not great with my colors, but this looks like maybe 5 or something like that, right? So we can move this. I don't know if this is 5 or not. You guys can tell. Right? He kind of looks like maybe this guy. Maybe this guy? I don't know. I think so. All right. Right? So we line them up. We basically cut the picture apart digitally. Historically, they'd have to print it out and cut it, right? Blow it up and you know, develop a picture of it, cut the picture out, put it back together, take another picture of it, copy it. Today we can do this digitally, right? We take a digital image on our microscope, cut those chromosomes out, sort them by size. You can apply stains to them. If you do this on a regular basis, you get to where you recognize the color patterns and the staining patterns, and you can identify the number based on staining patterns. The idea is you can line them all up. And you can see how many of each chromosome we have. Do we have the right or wrong number? Again, if I go back here, an aneuploid, which can be monosomy or trisomy, or it can actually be more than trisomy, is an individual where they have the wrong number of chromosomes because something goes wrong. There's a non-disjunction during the meiosis process, resulting in one too many chromosomes somewhere. Now again, this does not work well in most mammalian species for the larger chromosomes, but for some of the smaller chromosomes, it is a survivable condition. One of the most common ones is Down syndrome. We call it trisomy 21. It's where there are 3 copies of the 21st chromosome. Maybe you've interacted with, you know somebody with Down syndrome. There's been some actors with Down syndrome. What's that? They're very sweet, typically very sweet, well-mannered people. They can be an absolute pleasure to interact with. You're absolutely right. There are a number of, they are different. Right? They are different. There are physical differences and there are mental differences, as opposed to those of us that are non-aneuploids. Okay. There are also another chromosome. Again, if you have trisomy of 1 or 2, that individual is most likely not going to make it far past fertilization. It's normally a non-viable embryo. But 21st chromosome in human species, those individuals can survive. You can have trisomy or monosomy on the X chromosome. You can actually have more than one X chromosome, or only one X chromosome, and those individuals can actually survive. They're going to be different. There's going to be some developmental differences. Often there's going to be some fertility differences. It's pretty rare that individuals with the incorrect number of X and Y chromosomes are actually fertile. It's actually pretty rare. And they just tend to have this, again, they're expressed, their genes are expressed differently because they have more or less genes than is what is normal. Okay? So triplo-X, which I didn't find a name for the disorder, just simply triplo-X or trisomy X. Klinefelter syndrome, that is an individual that has a single Y chromosome, 2 X chromosomes. They will be male, they will present as male, but they will have more feminine qualities to them. And again, often, very rarely are they fertile, almost always they're sterile. You can have Turner syndrome where you have an individual with a single X chromosome. In humans, that presents as just difference in size and some different skin in some different areas, but various impairments. Right. How do we know this? Well, we can take karyotypes of a cell as it's dividing for the embryo and actually test it or measure it. And we could also take a karyotype of a cell. Doesn't have to be an embryo. It could be after birth. It could be in a mature individual. And again, I've kind of hit on this. Most of the chromosomes we can't do this with, but the X chromosomes in many species of animals, they can have the wrong number of X chromosomes and present viable individuals. One of the reasons for that, one of the reasons that the X chromosome is okay to have a different number of copies and still be viable is because in most cells, only one X chromosome is going to be active. So what happens is early in the development of a zygote, they'll reach a point at a few thousand cells where within each cell, if it's female, one of the X chromosomes will actually deactivate. And it will kind of compress, and it's harder for then to extract or utilize the information on that chromosome. It becomes what we call a bar body, B-A-R-R body. And so the bar body still is the inactive X chromosome within that cell. And so with that particular cell, the other X chromosome is the one that gets expressed. Whatever genes are coded there gets expressed. So again, this happens in most animals, around about 1,000 cells or so within the zygote. And then every cell, right? Early in embryonic development, yeah, that's what I'm saying here. Early in embryonic development, X chromosome in each cell is deactivated. That generates a bar body. And that bar body will remain inactive in every daughter cell produced after that. So at a certain point in the embryo's development, one X chromosome shuts off in that particular cell. And it's kind of random, right? This cell, one X chromosome shuts off, and the other cell, the other X chromosome shuts off. And then every cell produced after that will have the same inactivated X chromosome. So different cells within each animal is going to have a different X chromosome either on or off. What we've been able to figure out in cats, in domestic cats, what we refer to as calico condition, calico pattern in cats. What we found is the black and orange fur color is coded on the X chromosome. Okay? And so what happens is, again, remember, some cells one X chromosome presents, some cells the other X chromosome presents. And so if a cat has one orange X chromosome and one black X chromosome, some of the cells the orange X chromosome is active, some of the cells the black X chromosome is active. And so the cells where the black chromosome is active, that will present a black fur color, and the orange color will present an orange color. Confusingly, the white fur color is on a different chromosome. It's not on the X chromosome. That's not uncommon. That outward appearance will be controlled by more than one gene. Okay? Cats are no different. The white gene is on a different chromosome. It's also same thing in cattle. Red and black is on one chromosome, and presence of white is on another chromosome. That's not uncommon, a fur color. I think it's somewhat unique in cats, though, that the orange and black is on the X chromosome. So again, the reason you have this, what we call this calico color, where you've got splotches of orange and black, because some of the cells have an active X chromosome that's got black on it. Some of the cells have got an active chromosome that's got orange on it. The presence of the white says you've got white spots in particular areas. Now, interestingly, so based on that knowledge, why would calicos almost always be female? Here's where I check if you're listening. We'll maybe engage you a little bit on why is this important. So why do we not expect, what's that? So how many X chromosomes do we expect the male cat to have? Just one. So if the male cat's only got one X chromosome, he's either going to have orange or black. What do we know if he has both? If I've got a male cat that's got both orange and black on him, and white, what does that tell me about him? Here's the,
That he might have a cat version of Klinefelter syndrome.
exactly. That is literally it.
Issue.
Yeah, that is literally it. That is exactly what we have. Is you can have a male calico if this occurs. If the male calico has orange and black, the only way he's going to have orange and black is if some of his cells have got an active black chromosome, and some of his cells have an active orange chromosome, and that's only going to occur if he's got the wrong number of X's. He's most likely infertile. I'm just reading on this site here. I've included the link that I borrowed the picture from here because I read through. I was like, oh, that's perfectly good. I thought this was really good, reasonably scientific information. You're welcome to follow up on that. They cite that they tend to have bone density issues. The male calicos will typically have other developmental disorders. Right? So it's pretty rare, one, that you're going to have that autosomal, or I guess it's not autosomal. It's rare you're going to have that chromosomal disorder in a male calico cat. It's a rare occurrence, and it's even more rare that they're actually going to be able to produce viable gametes because they've got the wrong number of chromosomes. Questions, comments, complaints, so forth. Okay. So 13 again chapter 13 focuses on learning more about chromosomes, coming to this conclusion, this theory that inheritance occurs through chromosomes, which is still not accepted widely at this point, early 1900s, still not widely accepted. But we're continuing to do more and more research and looking into how these genes and traits are being inherited and passed on from one generation to the next. And you've got all these weird, you got these X-linked chromosomes, and you've got these shared dominances. Not everything follows Mendel's laws, so we have to kind of try to put all that together and figure out what we're looking at. All right. So I found some GIF images. I'm not sure why they don't loop. All right. So we can just kind of go back here. Some other chromosomal disorders. Again, most of the time, if your chromosomes aren't perfect, it's not uncommon that you have either severe developmental issues or it's just fatal. It's not a viable condition. There are some other things. So we talked about what happens when I have during meiosis. We don't get good separation on the chromosomes. We end up with the wrong number of chromosomes. There are some other things, and so again, just to back up, so force these GIF images to go again. There's some other things that can happen, right? So sometimes, again, remember these chromosomes are just long strands of DNA. There's just simply hydrogen bonds holding them in most places together. I'm going to rotate this down a bit. There we go. Most of the time, it's just hydrogen bonds holding them together from one piece to the next. So there are places that this can mess up. There's pretty good checks in place. Again, not the best GIF images, but hopefully they kind of get the idea across. It's not uncommon that other things can go wrong. Okay, so you can have a situation where part of one chromosome gets spliced onto another chromosome and vice versa. Right? So part B can move here and there. Okay, that can occur. You've got part of it flips, and then we have what we call paracentric and pericentric. Normally when you see the phrase peri, P-E-R-I, that's like perimeter or perimeter. Perimeter is the distance around something, right? So when you see the term peri, P-E-R-I in a scientific term, we're often referring to around or near. So perimortem means the injury occurred right around the time of death, right? It's right around. P-E-R-I is around. So here we're saying the pericentric inversion is where you flipped around the centromere. So you see this taper right here. They're saying the centromere is right here. And they're saying that the pericentric flip, they're saying it flips around the centromere. The pericentric means it's flipped somewhere not around the centromere. Text does, I think, a pretty good job talking about, are these critical, are these survivable when this occurs? It depends. Does it change the way that the genetic information that is flipped or moved, does it change the way it's being read? If it does, then yes, it could basically cause a particular gene, a particular protein being built off of that strand of DNA to be built incorrectly. And that could cause a condition that's not good. It could cause cancer. It could cause a number of things. There's also deletions, right, where entire segments of a chromosome gets deleted and just left out, and every cell after that's missing that piece. So again, there's a number of, chapter 13, we talk about what happens when things don't go completely right with the chromosomes. Okay, that's kind of section 13 too. Another common thing that's typically fatal for animals, especially mammals, we're a little more frail in terms of we need our chromosomes to be just right. Plants, there's a little more flexibility. It's not uncommon in plants we observe what we call polyploids. Polyploids means that you've got an entire extra set of chromosomes. And so for most cells, what we call somatic cells in humans and most animals, they're what we call diploids. It's diploids, 2 sets of chromosomes. Triploid, meaning you would have 3 sets of chromosomes. Quadploid, 4. So polyploid is anything more than 2, the wild type. Why in the world would you want that? Well, the genes, again, what happens when you have more than one chromosome present? Well, you tend to express more genes. You produce more of the proteins or the enzymes that are coded for on those DNA strands. If a cell has more copies of that chromosome, it just makes more and more of the thing that's being coded for. If I'm producing crops and I want larger apples or larger bananas or more hardy wheat plants, I might actually want more of the things that are being built inside those cells. So a lot of the crops that we consume, plant-based crops, they tend to be polyploids. We have selected for individuals that are more likely to produce, and when farmers were selecting for these, we didn't understand why this particular apple tree was producing a much better apple. We just knew it was. So we would pick that particular apple. And so, again, we observe this polyploid is actually beneficial to us as humans in a number of different organisms. Okay. So, again, we talked about chromosomal inheritance theory. Early 1900s, we proposed, hey, we think genes inheritance occurs on the chromosome. But you actually got in the late 1800s, early 1900s, a lot of people making the observation that, protein could actually be what's passing genetic information. And there's some pretty good arguments of, hey, the protein, we know there's lots of protein available and present in organisms and cells. That's pretty obvious to see. There are, in trying to relate it back to speaking a language or communicating from one cell to the next, there's 20 amino acids. So, well, that kind of makes some sense. Most languages tend to have, especially most romantic-based languages and Germanic-based languages tend to have anywhere from 20 to 30 letters in their alphabet. So it makes sense that when communicating information from one to another, I'm going to want a lot of possible combinations to make that communication. So the idea of 20 amino acids serving as letters, it lends itself to that's obvious. That's how organisms are communicating from one generation to the next. And we simply hadn't completely nailed down enzymes and other, what exactly do we need proteins for? What exactly do proteins do? We just knew there was a bunch of it. Okay? On the other hand, you've got scientists knew, right, kind of doing a simple experiment like what you guys did with the strawberry, where you extracted lots and lots of DNA, a big blob of DNA out of that small little strawberry, just with real simple chemicals, household chemicals, right? So we knew that there was this nucleic acid very prevalent in cells as well. So it also was a possible candidate for translating or transmitting genetic information. One thing that kind of weakened the argument was this idea, well, that's only 4 letters. There's only 4 nucleotides that we see present in nucleic acid. At least in DNA, there's 4. There's adenine, adenosine, thymine, cytosine, and guanine. We did notice, we did understand that in RNA and other nucleic acids, we had uracil in place of thymine. We understood that, but there's only 4 components as opposed to the 20 we see in proteins. Now, Chargaff came to a realization when he studied some nucleic acid. He came to this realization that the amounts of cytosine and guanine were the same. Right? So if he took a sample of DNA and analyzed its components, he found equal amounts of cytosine and guanine, and he found equal amounts of adenosine and thymine. So C and G were equal, A and T were equal. This is important, as we learn later. So we look in chapter 14, we're really focusing on DNA and how it's structured and what it's doing. Now again, trying to decide, is it protein or is it DNA? Which one is actually causing for genes to be passed from one generation to the next? Right, now, I went on the internet, I read through, I think your text does a pretty good job explaining technically what happened with the Hershey-Chase conclusion. This was the definitive evidence. They also talk about, I can't remember the individuals, but they talk about where they infected a sample of rats or mice with different kinds of bacteria, provided significant evidence as to that it was in fact DNA that's being moved, but there's some holes in it. Hershey and Chase took some bacteria and they infected it with a virus, right? So viruses, in the particular virus they used, had a protein coat, and then it's got, a virus has nucleic acid, okay? And what most viruses will do is they will inject that nucleic acid, as you see in this GIF here, they're injecting nucleic acid into the cell, and then the cell will take that nucleic acid and begin building the protein necessary to create a copy of the virus. So they'll take it, they'll hijack what the cell's doing. If you've got the Pfizer or the Moderna vaccine, that's actually containing RNA that's going into your cells, and your cells are producing the spike protein that's on the surface of the coronavirus. So if you encounter coronavirus, your cells have seen the spike protein, they know what to do with it. What Hershey and Chase did, and what a lot of this research at this time they were doing, they were using radioactive isotopes because they could trace them. So they could trace the radioactive isotopes. And so what Hershey and Chase were doing is they said, well, some of the proteins used to make the coat, the structure of this virus, uses sulfur. So they're going to inject a sulfur isotope and get the bacteria to use that sulfur isotope to make its own protein structure. Okay, and then they, in a separate sample, they injected some radioactive phosphorus into another sample of the virus. So the virus would use that radioactive phosphorus to make its nucleic acid. And they looked to see which one in the cells that were infected, did the sulfur make it to the cells that were infected, or did the phosphorus make it to the cells that were infected? And what they found was the phosphorus, the radioactive phosphorus, which was in the nucleic acid, that was what got passed on to future generations of the bacteria after they've been infected. The sulfur did not. Because there's nothing passing the sulfur from one generation to the next. The protein was not that the conclusion there is that protein is not what's causing the inheritance. Protein is not passing on the genetic information because the sulfur is not getting passed on from one generation to the next. The phosphorus, on the other hand, phosphorus is getting passed on from one generation to the next. That means the phosphorus, the DNA which is being built from the phosphorus, is what's containing the genetic code needed for the next generation. So groundbreaking, real important discovery moving forward is a protein or is it DNA? Well, this really kind of hammers that home. And as we proceed through chapter 14 and we take a really in-depth look at nucleic acids, especially DNA, really focusing on DNA. Discuss RNA a little bit, but really focusing on DNA. Okay, so review slide, we've seen this before. We talked about chemistry, we talked about macromolecules, proteins, lipids, nucleic acids, right? So deoxyribonucleic acid, DNA, this is the molecule that stores the genetic information. This is typically in eukaryotes, it's in the nucleus. Prokaryotes, it's in kind of a nuclear membrane. The structure, the basic structure, if we blow this up and look at one of these guys, right? The basic structure, and I've pulled some different graphics from the internet here. You've got your phosphate group. This one is colored that yellow. So your phosphate group, which is yellow in all of these graphics. This is adenosine. So this one has this adenine nitrogenous base attached here. So you got your phosphate group here. You have a 5-carbon pentose sugar. Now, come on now. I don't know why it gave me an extra line. I apologize. There we go. See, let's try this again. I may go to the board. 5-carbon sugar. This, to me, that appears orange. I'm hoping that's orange. The green adenine is over here. Right? And the carbon sugar, what is this, purple, blue down here? Okay, whatever color this guy is. The 5-carbon sugar, up here it's orange, it's this blue color down here, okay? This one is adenosine, but all of them share the same structure. You've got a phosphate group on one side of the carbon sugar, 5-carbon sugar. You've got a base, a nitrogen base, on the other side. They all share this same structure. Now this carbon sugar, and as we said, it's 5 carbons here. I'll go to the board. Actually putting numbers. Putting numbers on these is a little tough to do on the mouse, but this is normally your 1' carbon, 2' carbon, 1''s here, 2' is there. Your 3' carbon there. Your 4' carbon is the end of this, end of the pentagon there. Pentagon is completed with this oxygen up there at the very top. So in organic chemistry, what we normally do, because most of you guys are probably going to go on to take an organic chemistry class. When we're drawing an organic compound, we normally don't label every carbon, every corner with a C. Right? So if there's a corner and there's no letter there, that means it's a carbon at that point. Okay? We normally, very rarely, do we draw the hydrogens because there's just a bunch of them. It just gets really confusing if we put all the hydrogens. Same thing with carbons. If we put all the carbons, it gets kind of confusing. So normally don't put these. So where's the 5th one? Well, the 5th one is this corner right here. That is the 5 carbon. The 5' carbon is out there, and that branches off and goes to the phosphate group. Okay. So the phosphate for this particular nucleic acid is going to bind to this 5' carbon. Okay. The phosphate on the next nucleotide is going to bind to this oxygen attached to the 3' carbon. So your next nucleotide is going to be down here. You see that? One prime, 2 prime, 3 prime, 4 prime, 5. So this is important because, again, this backbone of the DNA is formed on the combination of the phosphate group linking to a 5-carbon sugar. That links to the next phosphate group, which links to the next 5-carbon sugar, which links to the next phosphate group, linking to the next 5' sugar. I'm sorry, 5-carbon sugar. So again, when we lysed DNA, when we extracted DNA from those cells, those strawberry cells in the lab, remember it's just this big long glob. That's meters and meters, because that's thousands and millions of cells we pulled all the DNA out of, and it's just making meters and meters of this glob of these long strands of molecules strung together. And this is the way they form, right? We bind to a 3-carbon, we bind to a 5-carbon, next to the next 3-carbon, next 5-carbon. And when you look at the other end, so on this end, we're bound to the 5th carbon on this side. On this side, it's basically flipped, right? So going in one direction, it's going from 5 to 3. On the other side of the strand, it's going from 3 to 5. I know it's kind of hard to see. There's a lot going on there. I get that. What we want to know, what we do want to understand is they're running on what we call an anti-parallel orientation. The strands are basically running opposite directions of each other. They are complementary. Remember, Chargaff's rule says that the number of A's is equal to the number of T's, the numbers of C's is equal to the number of G's. That's because they bind with each other, right? So this lends itself to try to, we still didn't understand, we didn't know exactly how DNA was built, what it looked like, what the structure was. We've just now, and we still can't see an actual DNA structure, like the single strand. We can see chromosomes. We're just now, and I think we may just now, I guess we're just now getting to where we can actually see it with scanning electron microscopes. We might be getting close, but it's hard to see this. We had to do the best we could based on the information. Now, because they're structures, we do know that, again, we know A and T bind together. We find that there's 2 hydrogen bonds that bind those, right? There's 2 hydrogen bonds that bind those, and the CG connection is bound with 3 hydrogen bonds. You can see that here. Okay. So pull this graphic from your text. A to T, 2, C to G, 3. Whatever, and rearrange that. I've not tried to make some mnemonic structure out of that. Rearrange that to something that's easy for you to remember. So that's kind of the takeaway there. All right, so how did we actually figure out what the DNA structure looks like? Well, we got to about the 1950s. We start really beginning to understand, we're starting to understand that again, we've got definitive proof at this point that the chromosomes are what's actually pushing genetic code. Just now starting to understand, well, how complex is this? If that's what's doing it, how complex is this? I read what your text put in, and I kind of did a little research. I'm like, I thought your text your text is accurate. I just thought, well, it's not as detailed. Maybe this helps a little bit. So everything I've got here is in your text. I just tried to line it out in the timeline a little bit. Okay. So you got Pauling, Linus Pauling, using X-ray diffraction, which that's another science. If you want to learn more about that, I'll leave that to you. But using X-rays to really look at protein structures, exactly how protein molecules look and how they're built. Okay. So later, Maurice Wilkins works with Pauling to apply the same techniques he was using to look at protein, using what he learned from proteins, trying to use that to look closely at DNA. Then you've got Rosalind Franklin, who really, arguably, is responsible for putting all this together. Right? Rosalind Franklin putting all this together, working within Wilkins' lab, furthers the research and really takes the X-rays and is able to apply some X-ray methodology, looking at and gets a pretty good idea what DNA structure looks like. And then building a little further on that, because this is how science works. Is one person makes a discovery, the next person takes that and goes a little bit further, the next person takes that and goes a little bit further. There's nothing wrong with that. So you have Francis Crick and James Watson, who are almost always historically credited with the discovery of DNA. Well, now, they found the definitive shape of DNA. They really made the definitive conclusions that have held up over scrutiny of exactly what does the DNA structure look like. It's a double-stranded helix pattern. It's a double helix pattern that spirals and rotates, and then that gets coiled up tighter and tighter. So Watson and Crick are often the ones we credit with the discovery of DNA. Well, it's the discovery of DNA structure is really what we credit them, who we should be credited for, what we should be credited them for. The Nobel Peace Prize, this is the 1950s. This is when they're really starting to put these pieces together. In 1960s, Crick, Watson, and Wilkins were awarded the Nobel Peace Prize for the discovery because Rosalind Franklin had since passed away. And at the time, Nobel Peace Prizes were not awarded to people that had passed away. I think they've changed that recently, but at the time frame, that was not the case. So she was not credited. But that's as best I can kind of reconstruct the timeline. I'm not a history person. But I thought, well, maybe that helps a little bit. To just kind of read what they wrote in the text. I thought, it's complete, it's accurate. I just try to put it a little more visual for you. Okay? All right. Thought question. Apply two sides. I'm just going to code on. By the way, we're going to learn later. We just code off one, right? Only one side is actually red, and protein is built off one side of the DNA. We know this. So why is there two? That's part of it. That's one advantage. And honestly, scientists aren't 100% sure that there's reasons, but that's definitely an advantage. What are the advantages? Why two sides? What advantage does that provide, knowing what we already know about biology to this point?
I just think everything's based off of a simple structure.
Okay, simple structure. In case one side's damaged, what else? How does this help with mitosis? What happens before mitosis can occur? What do we have to do? What happens in interphase? What's that? Yeah, we got to copy everything. Need two copies. I got two strands. I make a copy of each side. It's the same information. So I can just go right up the middle. Right? I didn't know how much time we would have. Guys, there's a really cool video here. I'm not even kidding. Go watch this. This one's not in your text. Okay? Best I can tell, this is all correct and accurate. We're not going to watch all of it. But I did want to skip ahead and look at some things here. Okay? So it's not actual. It's a representation. But I wanted to show you we talking about here? DNA replication, perfect. Exactly what we're talking about. So splits it into two halves, makes a copy of both halves.
Where is that at? Ribosome?
This is inside the nucleus before this is met. And they're saying that this is approximately what it should be going in real time. This is occurring in the nucleus during interphase. For, how do we know this is real time? Well, for knowing about the length of those chromosomes, right? So if we know about how long those chromosomes are, we know about how long it takes cells to divide. We've been able to watch that under a microscope. So we know about how long that chromosome, you can figure out your speed, right? You know your distance, you know the amount of time, so we can figure out the speed. And that's what I wanted to show you this to explain to you again. This goes fast. ATP production, protein synthesis, copying of DNA, it has to happen fast. And I thought this video was really cool because they do show you, in some cases, just how fast this is going. During interphase, to copy your DNA, that's how fast it's going. Right? So anyway, yeah, I know. I kind of chuckled on a few of these, and the sounds are just, I know. All right, guys, that's what I got. You guys have a good rest of your week.